Good day, this is Professor Will, CBMD PhD. Today is Friday, August 21st, 2020. It is 5:54 and 42 seconds a.m. Eastern Standard Time. And this is a continuation of my corona research. But before we get started, let's take a look at something I wrote. In today's video diary, I'd like to highlight that I've been switching up my routine a bit, such as reading Wikipedia, my recent topic being herd immunity. I actually started reading Encyclopedia Britannica way back in the mid-1980s, when I was just about 6 or 7 years old. I witnessed the pioneering of the ages at a great wavelength. In the year 1995, I discovered Encyclopedia Multimedia optical CD called Encarta 95. I decided to purchase the disc with my own work monies. I was really excited about this new style of encyclopedia, and studied on my computer for multiple years. My Encarta actually had a dictionary built in, which helped me to spell check my IRC text back in the fall of 1995. In further years to come, I was so excited about Encarta on disc. I had asked my parents to purchase me the latest Encarta 2000 for my birthday. It even had the online update option built in. I really enjoyed my time reading a digital encyclopedia. I find reading the digital version much easier on the eyes, with the screens lit up and adjusting font sizes. I was high functioning as a teenager and could easily talk circles around physicians. I was actually over, above, and beyond doctorate standards at the age of 16 years old. I had been reading encyclopedia for about 35 years plus, and I still find encyclopedia fascinating, which made an enormous intellectual impact on my life. Knowledge is the true wealth of the world, and it is quite phenomenal what you can learn. The possibilities are endless. I am pure educated. Well, I hope my audience enjoyed my presentation. Hope to see you again. Until next time, let's continue on with my corona research. Dilated blood vessels in the lung may explain how low oxygen levels in severe cases of COVID-19. Date August 20th, 2020. Source the Mount Sinai Hospital, Mount Sinai School of Medicine. Summary. A new pilot study suggests that COVID-19 is causing significant dilation of the blood vessels of the lung, specifically the capillaries. A new pilot study from the Icahn School of Medicine at Mount Sinai suggests that COVID-19 is causing significant dilation of the blood vessels of the lung, specifically the capillaries. This vasodilation is contributing to the very low oxygen levels seen in COVID-19 respiratory failure. It also helps explain why the disease behaves differently than classic acute respiratory distress syndrome, ARDS. In classical ARDS, pulmonary inflammation leads to leaky gut pulmonary blood vessels that flood the lungs with fluid, making the lungs stiff and impairing oxygenation. Many patients with COVID-19 pneumonia demonstrate severe hypoxia that is markedly out of proportion to the degree of lung stiffness. This disconnect between gas exchange and lung mechanics in COVID-19 pneumonia has raised the question of whether the mechanisms of hypoxemia in COVID-19 differ from those in classical ARDS. The discovery was serendipitous. Researchers were initially assessing cerebral blood flow in mechanically ventilated COVID-19 patients with altered mental states to look for, among other things, abnormalities consistent with stroke. They used the robotic transcranial Doppler TCD, the Lucid robotic system by Nova Signal, to perform a bubble study, which is a non-invasive and painless ultrasound technique. It is remarkable that a diagnostic machine used to study the brain could give us insight into the pathophysiology of a pulmonary disease. The benefit of using this particular system was that automated monitoring allowed providers to assess cerebral blood flow while minimizing the potential for exposure for COVID-19. During this study, agitated saline with tiny microbubbles, is injected into the patient's vein, and TCD is used to determine if those microbubbles appear in the blood vessels of the brain. Under normal circumstances, these microbubbles would travel to the right side of the heart, enter the blood vessels of the lungs, and ultimately get filtered by the pulmonary capillaries, because the diameter of the microbubbles is bigger than the diameter of the pulmonary capillaries. If the microbubbles are detected in the blood vessels of the brain, it implies that either there is a hole in the heart so that the blood can travel from the right to the left side of the heart without going through the lungs, or that the capillaries in the lungs are abnormally dilated, allowing the microbubbles to pass through. In the pilot study, 18 mechanically ventilated patients with severe COVID-19 pneumonia underwent TCD with bubble study. 15 out of the 18, 83% patients had detected microbubbles, indicating the presence of abnormally dilated pulmonary blood vessels. The number of microtubules detected by the TCD correlated with the severity of hypoxemia. indicating that the pulmonary vasodilations may explain the disproportionate hypoxemia seen in many patients with COVID-19 pneumonia. Previous studies have demonstrated that only 26% of patients with classical ARDS have microbubbles during a bubble study. Furthermore, the number of these microbubbles does not correlate with the severity of hypoxemia, implying that pulmonary vascular dilations are not a major mechanism of hypoxemia in classical ARDS. It is becoming more evident that the virus wreaks havoc on the pulmonary vasculature in a variety of ways. This study helps explain the strange phenomenon seen in some COVID-19 patients known as happy hypoxia, where oxygen levels are very low, but the patients do not appear to be in respiratory distress. If these findings are confirmed in larger studies, pulmonary microbubble transit may potentially serve as a marker of disease severity, or even a surrogate endpoint in therapeutical trials for COVID-19 pneumonia. Future studies that investigate the use of pulmonary vascular constrictors in this patient population may be warranted. The pilot study has since expanded to collect data from approximately 80 patients, including those with less severe disease, and will evaluate the severity of microbubble transit and how it varies during the course of the disease. COVID-19 patients who experience cytokine storms may make few memory B cells. Date August 20, 2020. Source Cell Press. Summary. The release of massive amounts of proteins called cytokines can lead to some of the most severe symptoms of COVID-19. A new study now suggests that high levels of some cytokines may also prevent people who are infected from developing long-term immunity, as affected patients were observed to make very few of the type of B cells needed to develop a durable immune response. When large numbers of immune cells release cytokines, this increases inflammation and creates a feedback loop in which more immune cells are activated, and this is sometimes called a cytokine storm. We've seen a lot of studies suggesting that immunity to COVID-19 is not durable because the antibodies decline over time. The investigators focus on germinal centers, the areas within the lymph nodes, and spleens where B cells, the immune cells that produce antibodies, differentiate. Differentiation and changes in antibody genes are required to build immunity to an infectious agent. When we looked at the lymph nodes and spleens of patients who died from COVID-19, including some who died very soon after getting the disease, we saw that these germinal center structures had not formed. Because the disease was so new, animal models for studying COVID-19 infection were not yet available at the time they began their study. The researchers instead gained insights from previous studies involving mouse models of other infections that induced cytokine storm syndrome, a malaria model, and one of bacterial infection in which germinal centers were lost. In people with severe COVID-19, one of the most abundant cytokines released is called TNF. In the infected mice, TNF appeared to block the formation of germinal centers. In previous cytokine storm models, when the mice were given antibodies to block TNF, or had their TNF gene deleted, the germinal centers were able to form. When the researchers studied the lymph nodes of patients who had died of the disease, they found high levels of TNF in these organs. This led them to conclude that TNF may be preventing the germinal centers from forming in people with COVID-19 as well. Studies have suggested this lack of germinal centers happens with SARS infections. We even think this phenomenon occurs in some patients with Ebola, so it was not surprising to us. The researchers also studied blood and lymphoid tissue from people with active infections who were in different stages of COVID-19. They found that although germinal centers were not formed, B cells were still activated and appeared in the blood, which would allow the patients to produce some neutralizing antibodies. There is an immune response. It is just not coming from a germinal center. Without the germinal centers, there is no long-term memory to the antigens. Researchers note that studies of other coronaviruses that cause colds have suggested that someone can get infected with the same coronavirus 3 or 4 times in the same year. The authors say despite their findings, they still believe a successful COVID-19 vaccine can be developed as it should not cause high levels of cytokines to be released. Mild COVID-19 cases can produce strong T-cell response. Date August 20, 2020. Source Cell Press. Summary. Mild cases of coronavirus disease 2019 COVID-19 can trigger robust memory T-cell responses, even in the absence of detectable virus-specific antibody responses. Researchers report. August 14th, the journal is Cell. The authors say that memory T responses generated by natural exposure to or infection with severe acute respiratory syndrome coronavirus 2 SARS-CoV-2. The virus that causes COVID-19 may be a significant immune component to prevent recurrent episodes of severe disease. We are currently facing the biggest global health emergency in decades. In the absence of a protective vaccine, it is critical to determine if exposed or infected people, especially those with asymptomatic or very mild forms of the disease who likely act inadvertently as the major transmitters, develop robust adaptive immune responses against SARS-CoV-2. To date, there is limited evidence of reinfection in humans with previously documented COVID-19. Most studies of immune protection against SARS-CoV-2 in humans have focused on the induction of neutralizing antibodies, but antibody responses tend to wane and are not detectable in all patients, especially those with less severe forms of COVID-19. Research in mice has shown that vaccine-induced memory T-cell responses, which can persist for many years, protect against the related virus SARS-CoV-1, even in the absence of detectable antibodies. Until now, it was not clear how SARS-CoV-2 specific T-cell responses relate to antibody responses, or to the clinical course of COVID-19 in humans. To address this gap in knowledge, researchers assessed SARS-CoV-2 specific T-cell and antibody responses in more than 200 individuals from Sweden across the full spectrum of exposure, infection, and disease. During the acute phase of infection, the T-cell responses were associated with various clinical markers of disease severity. During the acute phase of infection, the T-cell responses were associated with various clinical markers of disease severity. After recovery from COVID-19, SARS-CoV-2 specific memory T-cell responses were detectable. The strongest T-cell responses were present in individuals who recovered from severe COVID-19. Meanwhile, progressively low T-cell responses were observed in individuals who recovered from very mild COVID-19 and family members exposed to the virus. Our findings suggest that the reliance on antibody responses may underestimate the extent of population-level immunity against SARS-CoV-2. The obvious next step is to determine whether robust memory T-cell responses in the absence of detectable antibodies can protect against COVID-19 in the long term. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and computer science. I hope you enjoyed listening. Until next time, have a great day.